This is CheloniaCast, episode 55. Today we are joined by Dr. Robert Ossiboff, who is Clinical Associate Professor of Aquatic, Amphibian, and Reptile Pathology and Anatomic Pathology at the University of Florida College of Veterinary Medicine in the Department of Comparative, Diagnostic, and Population Medicine. There's a lot to dissect in this title and a lot of different sort of hats. I think it's a testament to the number of hats that Dr. Ossiboff wears and sort of playing this role as veterinary detective in looking at pathology of different diseases and sort of his specialty. He's published or written or co-authored over 80 different scientific papers in a variety of subjects related to his primary sort of profession of veterinary pathology and sort of disease dynamics. And he's also authored or co-authored 7 book chapters. And we're really excited to talk to him today. We haven't really spoken to anyone with a specific pathological profession and specialty. And so thank you for coming on, Dr. Ossiboff.
Thanks for having me.
All right. So I think we can kind of just jump right into things. So obviously you have a lot of knowledge about disease and infection and things of that nature. I'm kind of curious about some of your work with bog turtles and stuff, and whether you think bog turtles, obviously they're unique in a lot of ways, but if they have a uniquely higher risk of contracting herpes virus compared to maybe other chelonians or.
Yeah, so that's a great question. I did my bog turtle work a fair bit ago now, but I still work with bog turtles here and there. But my field bog turtle work involved bog turtles in the Northeast, New Jersey, New York, Massachusetts, and we were doing surveillance on them to try and understand what pathogens they might have. I mean, you both know that bog turtles are endangered. Their populations are declining. We don't entirely understand why all the populations are declining. And so whenever you have declining populations, investigating infectious disease is an important component of that. So we looked at bog turtles and then also wood and spotted turtles in the same habitats, and we screened them for a number of different viruses, one of them being herpes. And herpes viruses are it's a really interesting virus because herpes viruses themselves often don't cause disease in the host that they're supposed to infect, but when they spill over to related hosts, then they can cause really dramatic disease. So we screened them looking, kind of expecting to find something, but we were really surprised when we found that, more than 50% of bog turtles had herpes viruses, and it wasn't just one herpes virus. So there was a mixture of viruses in them, but the animals that had those viruses seemed to be overtly healthy. And that's kind of a theme that we've found the more that we look for herpes viruses in aquatic turtles, not just herpes viruses, also mycoplasma, is that it seems to be a pretty normal, typical infection for them. And we don't generally see disease caused by that infection. Now, that can change dramatically if you add stress into the system. So if you have a population of animals that is not experiencing stress, their environment's good, there's not much pollution, temperatures are what they should be, those viruses, most oftentimes are subclinical. All of us as humans have multiple herpes virus infections. They don't normally do much, but they can cause disease with stress. And so that's I don't think that the bog turtles are any more susceptible to herpes virus infection. I just think that, as you both also know, our studies of wild chelonians are really lacking. And because bog turtles are endangered and because there's more funds available to do their health studies, we were able to document that they had a number of different herpes viruses.
It's interesting. You kind of bring up an interesting question that I've sort of wondered about. When you say it's interesting to hear that you don't think they're more susceptible to the virus potentially causing an issue because you're assuming that virus has sort of evolved with that specific species of turtle for a long period of time, and there's sort of a mutualism that you wouldn't want it to become virulent per se and cause issues. But that brings a question about just in terms of species specific for turtles, immune responses and how susceptible they are to disease. I've worked with red-eared sliders and western pond turtles in Southern California, and it seems like there doesn't seem to be a lot of research on this, but that physiologically western pond turtles are a lot more susceptible to contracting infections. And from people I've talked to in that work with captive turtles, it seems like they're a lot, compared to red-eared sliders, that are a lot more robust. What do you think? Is that something that you've also noticed, or what are your observations with species-specific immune response and susceptibility?
Yeah, that's if there's one area that's really poorly studied in reptiles in general, it is their immune response and how they respond to infectious pathogens. And, there are certainly more robust species for and not just chelonians. We see this in other reptiles and even amphibians, species that are kind of in many ways bulletproof. They are resistant to many different things that would knock back some other species. And red-eared sliders are, a great example of that. I think that it, in some ways, it probably has contributed to their success in being able to establish themselves in so many different places where they're not necessarily native. It's just they're very, they have robust immune systems, and they don't succumb to disease as dramatically as some other species. You mentioned western pond turtles, and they do seem to kind of, we see a little more disease in them sometimes. There are a number of disease syndromes in western pond turtles. I was recently helping some clinicians up in the Pacific Northwest, and they characterized herpesvirus transmission from painted turtles into western pond turtles that caused fatal disease. And that's something that I had seen historically, when I was working in New York with map turtles, northern map turtles, same thing, And so that kind of gets back to the herpes virus, where, host adapted herpes viruses can spill over into closely related species and cause fatal disease. And that is, we've seen that in certain captive situations, and it's important information to collect because, particularly for animals in captivity and zoological collections or aquariums where, they're trying to create exhibits that show mixed species and highlight different animals, you have to be careful with the animals you mix because, some are much more sensitive, whether they be susceptible to disease, whether it be a decreased immune response, and that can result in health conditions down the road.
Yeah, so I guess when we're talking about things from like the perspective of wild chelonians, I guess I'd say I don't know. So I'm located in upstate New York, actually. So
Okay.
and I actually can't really name any populations of red-eared sliders that seem to survive or reproduce with our harsh winters and stuff like that.
Yep.
And so I guess that could be some amount of a limiting factor to red-eared sliders and bog turtles in my region interacting. And also just their habitat preferences seem to just not completely align. So I don't know how much of a threat red-eared sliders would pose to introducing these pathogens to bog turtles, but I don't know if you've noticed anything like that.
Yeah, no, probably not to the bog turtles, at least not in where you're part of their range. If you get more down into New Jersey, that might be a slightly different story. But yeah, it's an important point because you have to consider, since I've been in Florida, I've been much more involved in invasive species work because we obviously have an issue with that. And that, what the range of temperatures and conditions that these non-native species can tolerate is really important in determining the risks to the native species. And I guess, that's one good thing that the bog turtles up further north have is that they may not be exposed to invasive red-eared sliders as much. One of the things we did that, kind of getting back to some of the herpes transmission stuff is one of the things we did find in our bog turtle study up in the Northeast was there was one virus that was present in very low levels, but we detected in both spotted turtles as well as bog turtles. And when we see something like that, that's more concerning potentially for a virus that could cause disease. Luckily, we have not found disease associated with that virus. But, one of the challenges with any sort of turtle work, and you both probably know this, is when there's mortality events, you don't often find the dead turtles. You may find shells after the event, but you don't often find the sick or dying turtles. And that really limits what type of investigation you can do. I mean, there have been really good groups studying ranaviruses, which are very important pathogens of chelonians. And particularly for some of the box turtle mortality events, they've discovered that you can detect the ranavirus in the bone marrow in the bridge region of the shell, which means you don't necessarily have to have any soft tissue left. But with, decomposition of the carcass, with scavenging by raccoons and things like that, it can be really hard to truly document why there is a mortality event in certain chelonian populations. And I think that's one of the reasons why we don't understand some of the causes of mortality in certain chelonians as we do others. I mean, before we started recording, you were mentioning musk turtles or mud. If there's a mortality event in those species, you're not going to find them. And so that's one of the challenges, I think, that certain chelonians, certain aquatic, certain fossorial species really, it poses to us understanding not only their population numbers, but then when there's actually mortality events.
I've worked a decent amount in the Chelonian Research Institute, the collection, Peter Pritchard's collection, and it really is testament to what you're saying in terms of a lot of the specimens in there are all sort of artifacts at this point, but there isn't a lot of that, there's not, it's mostly fragmentary material in a lot of cases. They're just shells. And from thinking about from the clinical perspective, how much information is lost in that scenario when you just have a shell. In some of the cases we've seen, the shells will have abnormal sort of formations on them or something like that. And you wonder what caused this to die, how much information. It's still useful, obviously, now for other sort of areas of research, but you lose a lot of the soft tissue interest you can get there, which is really interesting. Maybe we can sort of step back a little and just talk about pathology at broad and what was your first experience working with a turtle or tortoise in sort of a veterinary capacity, and what did your training up to that point, and I'm assuming it was more dogs, cats, and typical veterinary work, what did that help you with understanding the turtle and tortoise?
Yeah, so it's a great question, and I think it's a good jumping point to start a discussion on things related to that. To be honest, I can't remember my first turtle or tortoise case in the veterinary medicine. I've been interested in reptiles and amphibians, since I was a young kid, and I didn't go into vet school knowing that I wanted to do what I'm doing now. But as you mentioned, in veterinary medicine, the focus of training is very highly the companion or agricultural domestic species. You learn about dogs, you learn about cats, you learn about horses, you learn about cows. And then once you get past those four, you start to learn a little bit less. And by the time you get to reptiles and amphibians, you learn marginal amounts. That is not the focus of a veterinary degree as it relates to getting passing your national board exam. And so most veterinary schools are training their veterinary students to what the majority of the veterinary students are going to practice. And there are certain levels and experiences that are required to be met both by the American Veterinary Medical Association, and then there's the National American Veterinary Licensing Exam, the NAVLE, that all vet students take, is also an important component. The students have to know the material to pass that exam. There's maybe one or two questions probably a year on that exam as it relates to reptiles. And so really, for veterinarians to get experience in reptile, amphibian, avian, there's more avian in the veterinary curriculums than reptiles and amphibians, but you have to take that upon yourself. You have to seek out externships. You have to you have to kind of get that knowledge. There's kind of as something to compare to, for people who get into veterinary medicine and they want to practice horse medicine, it's really hard for them to become a respected horse veterinarian if they don't already have a fair amount of horse experience before they get into vet school. And that's generally where, the people who want to get into reptile medicine or reptile pathology, amphibian medicine, you have to really go above and beyond to get those additional experiences. So I have to say, I cannot remember my first reptile or my first turtle case. I remember I had very early on in my training, I had a very cool bog turtle case. It was a captive bog turtle that had just necrosis of all of its limbs and its shell, and then these large masses that were absolutely filled with keratin. And this case would have probably been circa 2010, 2011. And my hope when I go, I'm returning back up to where I went to vet school at Cornell next month. And I'm hoping to actually pull the blocks from that case, because the hallmark histologic features of that case are what we now know as amidomycosis in different turtles. And so at that time, we had no idea of the disease. It would be very odd to see fungi present within a cyst with keratin. But that, I remember that case, and I remember the odd appearance. And if I can go back and look at it and I see fungi, there's a chance that I can do PCR on that now to determine if that was a case of amidomycosis back then. And so for those of you who don't know, amidomycosis is a fungal infection caused by a fungus in the genus Amidomyces. The best characterized fungus is Amidomyces testivorans. And that causes really dramatic shell lesions in a variety of different species of turtle. It was first documented in Western pond turtles that were being head started in captivity and then released out into the wild. So they had issues in some of those turtles. But in looking at collections, zoological collections for this fungus and the disease, it's been found in a wide variety of species. We in Florida have seen it in a free-ranging Florida cooter. So we know it's not just a disease of captive turtles. But we also know now that it's not just one fungus. So, initially when it was published, it was Amidomyces testivorans, and that's the cause of all of these lesions. We've done a lot of work since then. We know there's probably a variety of related fungi in the genus Amidomyces that can cause similar lesions. And that's kind of an area of active research for us, is to better understand, is this fungus a primary pathogen? Is it causing the disease by itself, or does it need other issues to then allow for infection and subsequent infection of the shell and the dramatic disease it can cause? So I would say that's probably my first. There was a Russian tortoise that had horrible metabolic bone disease, and then there was that bog turtle that had the epidermal inclusion cyst. And those are probably the first two cases I remember as it relates to a turtle or a tortoise necropsy.
That's interesting. The Amidomyces and sort of mentioning that. There was a paper by, I think Paul Gibbons is the lead author, and just talking about emerging infectious diseases in turtles. I don't recall if they mentioned the Amidomyces, but maybe that was prior to it becoming a well-known pathogen. Is that something that you would say would be added to that list if it wasn't initially on there as something that is concerning?
Yeah. And I know the paper you're talking about, and that's Paul Gibbons, Laura Adamovich, and Matt Allender, I think, wrote that second update of that paper. And I gave them a few pictures for that paper. I don't recall if Amidomyces is in that paper either, but yes, it is absolutely one of the, there's a number of emerging diseases of chelonians that we're trying to understand better. And that's one of them. That's a really hard disease, because that's a disease that sometimes you can look at the plastron, the carapace externally, and not have any idea of how dramatic the disease can be internally. Sometimes you either need to use X-rays, radiographs, or a CT scan to see how dramatic it can be. And I mean, one of the earlier cases that I did of that was a young little diamondback terrapin that if you just looked at the outside of that shell, you'd think, there's a couple of areas where there might be some change, but almost 80% of that animal's plastron and carapacial bone was replaced by inflammation and keratin. I mean, you could tell that when you squeezed the terrapin because there was very little structural integrity left, but just by looking at it. So it's a disease that can be hidden. You cannot see what's going on and it be very, very dramatic.
Right. That's really concerning. Maybe we can jump up a little because you said something, just talking about the fact that it takes a lot of added training to become an exotic veterinarian. And that's sort of, I think that has to be a labor of love to do that. I mean, you're talking about years of extra training and work. What first got you interested in pursuing this as a career and sort of as a passion?
So yeah, the passion has always been there. I grew up in southern New Jersey with lots of eastern box turtles around all the time, snapping turtles, different emetids, and then lots of amphibians, pine snakes. I always had an appreciation for reptiles. I had a few of them as pets, but I did not, I made a couple of changes in my life career. When I first started going to college, I thought I wanted to go into human medicine. I had a family tragedy as a child, and I kind of wanted to go into human medicine in that field to try and address that. But I was in one of those classic, weed out pre-med discussions where they say, look to your left, look to your right, at least one of those people isn't going to make it with you to the end. You have to really want to do this, and I understand the purpose of those lectures, but it really made me think that if I went into human medicine in that career, I don't think it would find it as fulfilling as something else. And so I decided to pursue veterinary medicine. And then I worked as a technician in a vet practice during undergrad to pay the bills while taking classes. And I enjoyed veterinary medicine, but I liked the hunt for disease, as you mentioned. I like to figure out what was causing disease, and then beyond that, I didn't get as interested. And dog and cat, seeing them for vaccines, and it wasn't as really as exciting to me. So I went into vet med kind of open into what to do, and then I had my first opportunity to do veterinary research. And I did some research over the summer between my first and second year in vet school and studied an emerging pathogenic virus. And I was hooked. Like, I really enjoyed understanding infectious diseases and how they interacted with the host. So I stepped away from the vet curriculum for a few years to do a PhD in virology. And it was kind of in that time that I was like, what do I want to do with my life? The first idea was I want to make money. And so if you're going into veterinary medicine, you're already kind of not going to make a ton of money, not as much as if you go into some other fields. And so there were a couple areas of vet med where you could make some more money, but I realized that wasn't going to give me the fulfillment I really needed. And I was like, well, what can I do? I was like, well, I have this passion for reptiles and amphibians. I have this passion for medicine. I have this passion for disease. And at that time, chytridiomycosis, the fungal disease of amphibians, was just really being well characterized, wiping out amphibians around the world. So I decided that I wanted to commit my life to understanding infectious diseases of reptiles and amphibians, and to really then hopefully help and make a difference. And so a colleague of mine that I was working with on those cat viruses, she was a pathologist, and she said, you should really consider pathology, because what you do is you look at the tissues of animals that have died and you can see the changes, and then you can identify if there's maybe a novel virus, a novel bacteria, a novel fungus, and you can take all of the training you have as a molecular virologist and really work to characterize those diseases. And so that kind of, set me on the path. So, we talked about, you don't get much reptile and amphibian experience in vet school. You get even less reptile and amphibian experience in a traditional anatomic pathology residency. It's all dogs, cats, cows, production animals. So that's, my time that I spent in training had to be kind of long, because after I did the traditional pathology work, I then had to spend time at zoos to get exposure to the reptile and amphibian case load, which was really what was most important to me. And so that was fantastic. But then I thought for a while that my dream job was going to be working in one of the big zoos in the US, because that's how I would get to see the cool stuff. And I did get to see some cool stuff, but even in a lot of zoos, reptiles and amphibians are kind of not as important as the big charismatic megafauna. They're, the elephant necropsies, the rhino necropsies, the large primate necropsies, it was, even though it was important, I felt like it was pulling me away from my ultimate goal. So that's how I ended up in Florida. Florida is a cool place to do reptile stuff. And I was able to start a new program that specifically looks at diseases of amphibians, reptiles, and then we also do fish and other aquatic species. And it also, Florida has long had a molecular diagnostic lab that's designated to the investigation of disease in zoo and wildlife species, with a heavy emphasis on reptiles, given the historical expertise of people like Elliot Jacobson and Jim Wellahan. And so I was able to step in and take over co-directorship of that lab. And it's been fantastic. We get lots of really amazing cases, and I've been able, some people would say that, I'm hyper specialized, I'm very specialized in reptiles and amphibians. And that in some ways that's true, but we're close to 20,000 species between reptiles and amphibians. I don't consider that hyper specialized. I consider that, we're just starting to scratch the surface of diseases of all of these species, and we'll never understand all of it. But that's what it was. I love those species. They needed help, and I felt that I could contribute in that way.
You highlight an interesting point too, I think, in all of that we sort of talked about this, so that there seems sometimes to be kind of this schism between, with the age of Google and that sort of thing, when people come in with a perceived issue with their pet, they're very attached a lot of the time and will do some kind of self-diagnosis. But within reptiles specifically, it seems like a lot of the experience that you're having is actually creating new knowledge for the field. And so this is a field where particularly experience is very important, and you can't necessarily get that knowledge from a Google search, that sort of thing. Would you say that it's right to say that a lot of the field is building on itself just through experience and cases?
Absolutely. I mean, if you compare where we are in reptile veterinary medicine to other species, I mean, we are decades behind. There's so much that we still need to learn. If you take your dog or cat to the vet, let's say your dog or your cat has pneumonia, you take it to the vet. What we call them differential diagnoses, and those are the different things you would consider that can cause pneumonia in that species. And you take your dog in, and you're going to have a list that's 10, 15 things long. You take your reptile in, depending on the species, there may just be one or two things on that list. Does that mean that there's only one or two things that cause pneumonia in that species? No. It just means we haven't figured out all of the other things yet.
Yeah, I guess in that on that topic, when you evaluate, we'll keep things in terms of Chelonians. When you typically do like a health check on a turtle, is there any like very specific, I guess, like what's the typical procedure? And then I guess when things like blood work come into the equation, are there any very specific red flags you look for when trying to make some sort of diagnosis?
There a lot of times in vet med, a lot of our diagnostic attention is paid to a physical exam, right? We palpate, we examine everything, and the shell limits that immensely. And so we're kind of at a disadvantage in that. And so the one, some of the main things that we absolutely look for on external exam in turtles and tortoises is the head. The eyes can tell you a lot. The nares can tell you a lot of what's going on in the nasal cavity. And as much as they're not happy about it, you have to look in their mouths. The tongue can tell you an immense amount. Their esophagus can tell you an immense amount. A lot of diseases in those species manifest some way in the oral cavity. And so the oral cavity is an absolute important part of that. Doing what palpation you can from the axillary and inguinal regions is important. But then, blood work's really important. But, kind of with the blood work, we're also at somewhat of a disadvantage as well, because there are quick in-house units that a lot of vet practices can have to help them analyze blood work of domestic species. Those become a lot less reliable and fewer options available when we're talking about reptiles, including turtles and tortoises. And even looking at blood smears from those species, you need to have someone who knows how to count the different cells, because the automated counters that we often use in veterinary medicine mess up in most reptiles. And so when one of the things that we do in automated blood counts, and that's the quick way of saying, does this animal potentially have an infection? Are there too many white blood cells present? Are there not enough red blood cells present? One of the major cells that we look for are lymphocytes. Lymphocytes are small white blood cells that are important in a number of disease processes, but because reptiles don't have platelets, they have another cell type called a thrombocyte that has a nucleus. The thrombocyte and the lymphocyte look very, very close to each other. And you need to have a lot of experience to tell those two things apart. And most of our automated machines can't tell them apart. So you could get spurious information from some of the standard testing equipment we have on turtles, tortoises, snakes, lizards because of that. So again, it's one of the limitations, and I think it's one of the challenges that we have with people who are doing their best and bringing their animals to veterinarians who might not be as skilled with exotic species, but they really, really want to help, and that's a positive thing. But I think that's some of the reasons sometimes that reptile owners get frustrated with veterinarians, because it's a challenge sometimes for the veterinarians, especially if you're not in a really urban area. You might not have a highly skilled reptile veterinarian to take your animals to. And so that, it becomes a challenge. And so, we're always working on education. We're always trying to teach veterinarians. But for me, things that kind of give red flags, obviously, are, turtles or tortoises that are really underweight. Like, it's, and again, that's another thing, you can't see the fat on a turtle or tortoise very well. So you have to know what the weight of that species should be. And, for those that work with these species, you pick up a tortoise and it's not fat enough. It just feels light, but that's something you get from experience. So you have to feel, is it in good body condition? Is it starting to lose body mass? What does the oral mucosa look like? And then what does the blood look like? Kind of the big things, with any, and then for a lot of the common diseases, anytime the uric acid gets thrown off, and that's what's indicative of gout, especially for the tortoises, that's always a big red flag as well.
Yeah, I think you bring up a good point about a lot of times there just simply aren't those resources to get any sort of medical evaluation of captive reptiles. I mean, that's something I lived. I had a bearded dragon. I think she passed away in 2021, but she had some sort of tumorous growth or something when you palpated her stomach that were hard and asymmetrical feeling. And then she had a tumorous growth on her left eyelid that eventually got bigger and bigger. And I think what happened is it pressed on her brain and caused a seizure. But I still, to this day, can't really pinpoint exactly what it was. And there's people who would see reptiles, but nobody who specialized enough to really do anything about it.
Yeah, that's hard. There are, if you're looking for a veterinarian, you absolutely, the Association of Reptile and Amphibian Veterinarians, or ARAV, ARAV.org, ARAV.org, has a Find a Veterinarian feature. And that will allow you to find anyone who's registered with that group. Now, that doesn't mean that individual is going to be an expert in all of those species. It just means that they are a member of ARAV and that they're listed. So you sometimes have to do, a little bit of recon to find out who might be good for you. The hope is always, though, that any veterinarian you go to is wanting to do the best, and they will reach out and consult with others that may be experts if they don't have the information. And so I receive tons of consults via email all the time, and I try to respond to them as much as I can. But for example, your bearded dragon, right? So was it an older animal?
It was, I think, like 7 or 8 years old at the time.
Yeah. So 7 or 8, you're starting to get older for bearded dragons, and you said you thought there was a mass in the stomach, correct?
There were several, and they weren't perfectly round like eggs or something.
Yeah. So one of the most common tumors we see in older bearded dragons are gastric neuroendocrine tumors. And what you describe is the hallmark of, and it's probably has to do with the limited gene pool in bearded dragons. And they get these tumors, and they get them commonly, and they're one of the most common causes of acute death. And you said it had seizures?
I just say that because in her dying moments, she kind of like spazzed out a lot and had a lot of
Okay.
twitching and stuff. So I assume it had something to do with that. But
Yeah, because they also get alterations to their blood glucose with those tumors, and that could have caused it as well. But yeah, and then it's all about experience. The more you see, the more you will know. And I don't want to talk bad about veterinarians who are seeing exotics in areas where there are no exotics veterinarians. But I do understand sometimes the frustration of reptile owners with veterinarians and veterinary medicine.
Yeah, it's really interesting to just kind of harking on the experience aspect of things. And I do a lot of different math work and that sort of thing, and it's like it's kind of a different thing, but there is some relationship between. There are a lot of patterns in math that in the beginning of doing it, if you don't have a lot of repetition, you're not necessarily going to see it that fast, and you may have to work through all steps of the problem to figure it out. But once you sort of get a handle on it, you'll start to see broad patterns and things become a lot more, and then you can do more with that. So it's kind of an analogy, I guess, to experience in itself kind of breeds knowledge and ability to solve problems. So it's something that is important for sort of veterinary work with exotics. I mean, it's always fun to have these conversations because it's testament to how related all things in health are. We kind of jump all over the place, and there's so many linkages between all the different fields and subjects. Maybe we can return and kind of center on pathology, though, because that's one of your primary interests, and what we've talked about is all sort of related to that. But I'm curious how you define the pathology as sort of a word, because there's a lot in that definition.
Yeah. So, and this, I think, kind of goes, to flip back to a question that Paul asked before, about if animals come in and what I look for. So, in all honesty, I am a pathologist. So most animals that come to me are dead or on their way to being dead. So pathology is the study of, or anatomic pathology is the study of tissues for the process of disease. And that's done either via biopsy. So, if an animal has a mass and a sample is taken and submitted for microscopic evaluation, or if there's an animal that has died and needs a full postmortem examination, or as we call it in veterinary medicine, a necropsy, that's my job. And so I, am trying to determine why an animal is either sick or why it's died. And I do that by a combination of the way all of its tissues look to my eye when I do the exam, and then the way that all of the tissues look under the microscope, and what deviations are there from normal, and do those deviations from normal indicate a benign age-related change? Do those deviations from normal indicate an inflammatory or infectious change? Or do those deviations from normal indicate a potentially neoplastic or cancerous change? And there's other things we look for as well, but that's kind of the big answer to your question. But pathology is some of my specialty. I did additional training for that, but my pathology also involves molecular pathology. And so molecular pathology is then when we look at things under the microscope, let's say, so I'll use a couple of tortoise examples. So we look at things under the microscope. Let's say we've got a large tortoise and it has lesions in its oral cavity. I get some of that tissue, I put it on the microscope. If I see small to moderate numbers of parasites present within the cells and particularly in the nucleus, that tells me that's most likely a case of intranuclear coccidiosis. And I can confirm that with PCR. But that's I can see in the tissue the parasite, I know what's going on. But if I look at that tissue and there's mononuclear or lymphocytic inflammation with necrosis, and there's nothing else, then that could be a change that is typical of a ranavirus infection. That could be a change that's typical of a herpes virus infection, or that could be a change that's typical of, another type of viral infection that we don't yet know. And so I also do then the next steps, which is running the PCRs, either targeted PCRs or having next generation sequencing run to look for the pathogens that might be causing the disease. And so I use both of those arms, both anatomic pathology and molecular pathology, to identify and diagnose why animals may have passed. And then to me, the most important thing is using that when there are many animals that have passed, not so much to diagnose disease in a one-off individual, but trends of infectious disease that may hurt either captive or wild populations of species.
Right. So you have to be good at a lot of different things, the visual aspect, the quantitative aspect. It's sort of a holistic thing, which is exciting. I mean, when it comes to working with turtles and tortoises, pathology of turtles and tortoises, what are some of the key? They're so unique physiologically in a lot of ways. So I can imagine there's some difficulties that are unique to them in studying the cause and effects of disease. What are some of those challenges?
They do have unique physiology, and they have, some unique anatomy that can make it challenging. It's always, even just getting into some of these animals is a challenge. Just yesterday, we was doing a necropsy, unfortunately, on a huge alligator snapping turtle. It was really sad to see that animal perish, and it was a healthy, fat male that had vehicular trauma, unfortunately. But it's, when you're dealing with a 70-pound alligator snapping turtle, getting into the brain case of that skull it's a lot of work. You have to know the anatomy of the skull to actually safely remove the brain, because if you don't know that, then you can destroy the brain. And things like that are important because, we recently just published on a novel group of viruses that we found that caused inflammation of the brain in a couple of species of marine turtles, but also alligator snapping turtles. And we know little to nothing about disease in alligator snapping turtles. And so if the person that's doing a necropsy, one, doesn't know how to safely remove the brain, or to two, even remove and examine the brain, that's all information that can be lost. The other thing, turtles and tortoises can kind of make things a little harder is, especially for the wild ones, is they have tons of trematodes. They have lots of spear orchids or vascular trematodes, and those spear orchid eggs end up in all of the different tissues, and they cause really mild inflammation, but it can kind of disrupt the normal architecture of things. There's, with all the species, there's challenges, and there are some in turtles and tortoises that are unique as well.
Just out of curiosity, I know you're in Florida, so you get all three lineages. Do you know where exactly that turtle came from, or I guess which of the three it was?
I need to look into that a little more. It was probably one of the Suwannee ones, but I'm not 100% sure on that one.
Yeah. Cool.
Yeah, that's an interesting observation. They don't come out. I'm assuming a nesting animal, if it was a vehicular issue. But
it was a big male, so I'm not sure. I'm not sure why it was out, and it was sad because we see a lot of turtle vehicular issues, and most of them are females, like you said, nesting, and they get run over and their shell gets crushed. But this animal did not get crushed. It looked like it got caught under a vehicle and then drug, and it dragging against the asphalt wore down all of it. It was a very sad case. That animal ended up having to be euthanized because of its wounds, and not die on its own. And that it was in pain.
One thing that I wonder about too, just in reading some papers and just in general in science, is sample size can be a problem. Is that something that's a particular issue in sort of in what I've looked at in a lot of cases? I mean, just sort of logically, you'd think that you're describing a situation where your sample size of just different patients you can have and different physiologies, and it's immense, and that can be overwhelming. I mean, like you said, you have to know the specific anatomy of that organism really well to get the right kind of diagnosis, or ability to get the diagnosis. Um, in sample size, I mean, if you've got one individual for something, how do you sort of combat the sample size issue?
That's a really important question, and the main way we combat that is collaboration. You have, you can't work as an island. You can't, I have really good friends and colleagues that do similar things to what I do, and when I see something, I reach out. So just a few weeks ago, I had a really interesting spotted turtle case, and I've done a few spotted turtles. I'd never seen this before, but I don't have enough to say that. So I reached out to a couple of colleagues, and they had many, combined, we had lots of spotted turtle necropsies, and we had not seen anything like that before. So again, it's with science, it's always science is best in collaboration, and that is in all aspects, including veterinary medicine. But it makes it really important to then also be careful with how you interpret studies. So, for example, we were lucky enough to have funds to do that multi-year, multi-state bog turtle disease or bog turtle pathogen survey. And we found all of those different viruses. But if there would have been a small scale bog turtle mortality event, and we had one bog turtle carcass that we found, and we did not have that historical or healthy turtle data, and we ran a herpes PCR, we would have found herpes virus in that animal. And then you run into the issue where sometimes disease is associated with a certain agent, and it's not a true association. And so that's where sample size can also be really important, is when you're looking at disease in just one or two animals, you have to be careful saying what we found caused disease when you don't have enough baseline data.
That makes a lot of sense. In terms of, I always am curious with people that have written book chapters. A lot of the time, it's sort of a meta-analysis in the sense of for like large reviews. And so you've done a few of these sort of things, and specifically a few chapters for Jacobson and Garner's Pathology, reptilian pathology book, and effectively a textbook. I'm curious what you learned in the process of doing that and writing those pieces, and what did you learn just through literature or preparation for that?
I mean, I think, that's the cool thing about what I do, is I learn stuff all the time, and we don't, you learn, what you've been taught, and the things that you've discovered, but we don't all have all the time in the world to go back and do deep literature searches into what's happened historically to truly understand all of these things. And that's one of the places where, textbook chapters, while they do take a lot of time, they give you the opportunity to do a deep read into all the historical data that's out there that you might not have known about. And that also happens every time we publish a new paper on something we didn't know about before. I was involved. There was we found a novel virus. For a little while, it was called a bunya virus, but it's not a bunya virus. It's turtle Fraser virus, as it's been called, and it caused a mortality event in soft-shell turtles throughout Florida. It's still ongoing, and then there's other species affected as well. But as we were working that up, one of the veterinarians we were working with, she did a little bit of diving into the literature, and it turned out that in like 1980, someone had found a soft shell turtle that was sick, and they took some of its brain tissue and injected it into the brain of mice, and it caused disease, and they did electron microscopy on it, and they saw viruses that they thought were Bunya viruses, and it's probably the same virus that had been present, way back then, and it's just in the annals of literature, and you can't. There's so much data out there that the only way to really come up on it is to do really good literature searches as each topic comes up. And a textbook chapter is like a forced way of getting into some of that information.
Right. Yeah, it's fascinating. When you think about the specific variability, because you think about it at the species specific level, how susceptible different species are to different pathogens and the chance for sort of causing disease. We've talked about that a bit, but then at the individual level, there's also some level of how does that individual respond versus another individual. How much does individual variability play into the ontogeny of disease in turtles and tortoises and microbiome, that sort of thing? I mean, how does that factor into that?
I mean, it factors in a lot. And especially when we're talking about captive animals, one of the areas that we don't give as much attention to as we should is genetic variability. How much heterozygosity do those individuals have at different loci that are going to help them be more resistant to certain features of disease? And then so that's the inherent, innate aspect of it. But then you also have the kind of the acquired aspect of it, in that the husbandry for these different species is absolutely going to affect their ability to respond or develop certain diseases. We know that suboptimal conditions in terms of how they're living is going to result in stress. Stress cannot always be directly measured, but we know that stress is going to usually be associated with cortisol levels, increased cortisol levels, which are then going to be associated with decreased immune function. And so there's so many minor things that play into what can make one individual more susceptible to disease than another, that in some ways we have control over, but in many ways we don't.
Right. And yeah, it's interesting too. I think a key thing about a lot of your work is you've done a lot of next level analyses. I think a lot of, just in the past few decades, with the advent of really exponential increase in sequencing technology and ability to pinpoint specific targets and genes and that sort of thing in all aspects of disease development and susceptibility has been really useful. And you've done a lot of this work. I'm curious, I've thought about this a good deal, and we do a research project in Florida and have seen a lot of cooters that have really significant shell damage, and it seems like they heal exceptionally well. And so that raised a lot of questions about the sort of turtle and tortoise immune system and how that functions. Curious if you have any specific examples of sort of molecular adaptations that may confer upstream advantage to just that overall robustness to that overall immune function in turtles and tortoises.
That is a fantastic question, and I'm sorry to say that I don't have an answer for that for you, because science is so complex, and I definitely look at things from a pathogen perspective. Most of my research is trying to understand the pathogen and how that then can affect the host. There's an entire field on the other side that is looking at the host as it relates to, as you said, changes in genes, and that is an area where there are some people doing some great work, but much more needs to be done in reptiles. More work has actually been done in amphibians in that regard than it has in reptiles, particularly with MHC loci and chytridiomycosis, and we absolutely need to get that on the reptile side of things. The challenge whenever it comes to science and reptiles is there's not a lot of public interest in giving money to do this reptile research. For those of you that aren't aware of how you get funds for research in academia. The largest source of funds is applying for grants, and then you write a grant proposal, and that will give you money for those. The big granting agencies are the national granting agencies. So you have the National Institutes of Health, and they are probably the largest, but they don't care about anything that's not human related. You have NSF, which is the National Science Foundation. They will in some ways fund studies for reptile studies, but it has to have a certain theme to it. And then USDA is almost always about agriculture and production, and we don't have a lot of production reptiles in North America. We put a proposal in recently to try and do some work on alligators, because in some states alligators are a production animal, and it was not, we didn't get funds. And so really the funding for reptile research is incredibly limited, and there's just a few small foundations that do it. But this is the one place where people, like hobbyists and people absolutely interested in these species can make a difference, because TSA, Turtle Survival Alliance, they're one of the most reliable places where every year, you can, there are small grants, but you can do work with that. But that's one of the big challenges especially for the immune genes and the inherent parts of reptiles, is it's really, really hard to get money, because you somehow have to convince someone that it's important to study this. And a lot of times the only way that importance comes across is if you can tie it to how much money that species is worth.
Yeah, just to take things back to the immune system, I was thinking about what Michael was saying about the cooters. This is one of the cooters in our project that was hand captured. I don't know how clear that image is.
Oh yeah, lots of shell. Yeah.
Yeah, but you can see it had a apparent respiratory infection. It was kind of wheezing and stuff. So it was handed over to a vet. But one thing that was particularly interesting to me is the algae and stuff on the shell. It very clearly hadn't been basking very much, and I wasn't the one who caught it. I can't really speak to its activity levels, but just something I noticed.
Yeah. And they can have crazy shell lesions. I mean, a colleague of mine, Shane Boylan, who's with at Jekyll Island at the Sea Turtle Center, he does work with a bunch of different species. He recently sent me amazing shell lesions, and it's absolutely unclear what caused them. Like what is happening at the shell and the epidermis of these species is absolutely an area where there needs to be active investigation. And it kind of goes along with, what we mentioned with Amidomyces before. Amidomyces, we don't know. We haven't done infection studies, or they haven't been completed yet. It's possible some of these fungi just live on the surface of the shell, and it's some low-grade damage to the shell and the epidermis that allows them to gain a foothold and then get in, and that sets off a chain reaction. So there's so much we can learn about those species.
No, yeah, that's a really interesting idea. I could see that. I mean, just little chips and that kind of thing. If they just get a little foothold, there's sort of this race to see who can outplay the other. Can the turtle stave off the infection, or can the infection infiltrate or pathogen, I guess, or microbe, even fungus, whatever. But so maybe we could go into one area that doesn't get as much public attention, I think, sometimes, because it can be kind of labeled as not as interesting and more technical, but is the sort of technology that underlies producing results and inferences about what causes a lot of infections. And these kind of things could be, I mean, you mentioned microscopes and sort of like transmission, electron microscopy, that sort of thing, or even at the molecular level, like PCR. And that's something in perusing your work, you use PCR very frequently. Maybe you could talk a bit about some of the technologies that you use to make inferences and draw conclusions about some of these diseases. What maybe some stuff that's exciting to you that you're starting to use and that has some potential future applications.
Yeah, absolutely. So, PCR it's been around for a while now. It's an incredibly powerful tool. If you were to go back 30 years ago, people doing PCR of a single gene and doing sequencing of that gene could constitute an entire doctoral thesis. And now, PCR is something that I'll start undergrads doing as small research projects. So it's something it's very useful. PCR itself has grown. Conventional PCR is you run the PCR and then you need to run an agarose gel, look for a product of the right size, send that product out for sequencing. That requires you to kind of know what you're looking for. You have to have primers that are going to match the target you're hunting for. We then have, quantitative PCR is kind of the next step of PCR. And that is, you no longer need to do sequencing because you have primers, and then, some of the most reliable ways use a third piece in that reaction that is a probe that then matches the nucleic acid sequence you're looking for, and that's tied to a fluorescent signal. And when it's present, you get a burst of light, and then the computer system will measure the amount of burst of light at the end of each cycle and determine how much of what you're looking in is there. But again, you still have to know what you're looking for. I think, we're quickly moving to the point where next generation sequencing is becoming affordable, and it will be the next step. And that is super exciting because you don't have to know what you're looking for. You take a sample, you load it on, it analyzes everything, it gives you an output, and then you can look through that output to find what's present in it. But there are still limitations. So I mentioned that turtle Fraser virus study that we did. When we did the first next generation sequencing on those turtles, we didn't get an answer because the virus that was in the turtles was so incredibly different than everything that had ever been found before. It didn't match up to anything. And it wasn't until there was actually a scientist who had cultured the virus 15 years before, and there were still samples in a freezer, and they did next generation sequencing on that sample, got the whole sequence, and then compared it again to what we found in the turtles, and it was the same thing. So next generation sequencing is not the total answer because you can still miss things. The other thing is that if I want to, in almost any sample, I can find a virus. I can find a bacteria. They're everywhere, right? The important thing is to then take that data and bring it back to the animal. So one of the techniques that I use a lot, because it's pretty straightforward, is a technique called in situ hybridization. And so in that, it's similar to, so some people may know, there's a technique called immunohistochemistry, and IHC or immunohistochemistry uses an antibody against a protein to look for that protein present in a tissue. The problem is to do that, you have to have isolated the protein. So that usually means you have to culture the virus, or you have to have more information. In situ hybridization takes that issue away. So I'm going to, we can use the turtle Fraser virus as an example. So, we have diseased turtles, and we've taken some of the tissue from their brain, and we've subjected it to next generation sequencing. And now we've found this virus sequence. But we can find viruses in a lot of places. It doesn't mean that it causes disease. A good example of this most recent is and some of you may know, there's a disease of sea stars called sea star wasting syndrome. And some of the initial studies in that sea star wasting syndrome tried to ascribe it to a certain virus because they found that virus in next generation sequencing. But later studies showed that virus wasn't doing anything at all. So we don't want to fall into that trap. So in situ hybridization says, okay, we know the sequence of that virus you found in the tissue. We're now going to make a probe that matches that viral sequence. And you put it on the tissue, and it's labeled with a red color. And that says, so if there's inflammation in the brain, I should see that virus mixed in with that inflammation in the brain. And that's what we did. And that gives us more confidence that what we found was actually potentially associated and causing the lesion in the brain. The next steps beyond that, you really have to isolate that pathogen and do experimental infections in animals to replicate the disease.
Right. And so in your capacity as sort of a research professor, and you're doing a lot of. It would be different from someone who's sort of in a practice per se, and just you wouldn't necessarily be developing the technologies that underlie a lot of this, that make a lot of these processes possible. But you've got experience developing the primers and the antibodies, the things that are necessary for isolating those proteins and that sort of thing. What kind of work goes into that? And perhaps something that I think relates actually to people that go and see a veterinarian, because a lot of times samples will get sent off to a laboratory like a University of Florida, and there's a cost involved with that. And so is this something that's cost prohibitive, and where is that? I guess where is the cost coming from, and what's the future of that? That's a lot of questions. The first one though, what's it like to develop the primers and sort of the antibodies and things necessary for those techniques?
I mean, that's one of the things that I really enjoy, and one of the places where I feel like I can help the most. I have that training from my PhD. I've got a lot of, I did a fellowship in molecular pathology. I have a lot of experience in developing, primers and things like that. For the antibodies, that's an even harder one. And so one of the, I mentioned I did my PhD studying a virulent virus, but the virus I studied was of cats, nothing to do with reptiles. But, when I was studying my virus of cats, there were cell lines, which are cells that have been established from living tissue that you can grow in tissue culture in a flask in an incubator, that you can then use those to do studies on. And so there's a couple different cat cell lines. There's a couple different dog cell lines. There's lots of mouse cell lines. There's tons of human cell lines. Do you know how many reptile cell lines there were when I started?
You could probably count them on one hand, I'm assuming.
Yeah, one hand. So there were about 4. There's a box turtle heart cell line. There's a viper heart cell line. There's an iguana cell line that's not entirely reliable. And those are really the major reptile cell lines. And so, how are you going to characterize widespread disease, even if we're just talking about turtles and tortoises, if you only have a terrapene cell line? And so one of the things that we did early on was we established a large panel of reptile cell lines, many different Chelonian cell lines. We have Galapagos tortoise cells, Indian star tortoise cells, soft shell turtle cell. I mean, that's what you really need then to grow these pathogens in culture and do the next steps to grow, get the antibodies established, make the assays. And that's, it's kind of, as I mentioned before, we're far behind in the reptile medicine disease research arena, and we need to pull ourselves into the next century. And so that's, with the training I've had and the skills I've developed, I feel like that's one of the ways I can contribute. And that's kind of what I do. So, to then segue into your costs question, that, there are inherent costs in all of, this type of work. Good thing is primers are cheap. It costs like $4 or $5 to buy a primer. It's a cheap thing. You need to do trial and error with different primers to make sure they're working. But primers aren't a huge cost. The reagents need to run the PCR. They make the cost go up a little bit. Paying the person to run the PCR is one of the big costs that kind of factor in. But the important thing, I think, it relates to cost. And so I'm co-director of the Zoological Medicine Diagnostic Lab at the University of Florida. We are not the cheapest lab out there. We're not the most expensive, but we're not the cheapest lab out there. But I go above and beyond to make sure our costs are as cheap as they possibly can be for the client submitting tests to us. But so let's say, Mike, let's say you had a tortoise, right? And you wanted to test that tortoise for intranuclear coccidiosis. So you send a swab to me to test the tortoise for intranuclear coccidiosis. That one swab, I need to run at least 3 tests to test that. I need to test the swab you sent me. I need to have a negative control to make sure that there's no inappropriate amplification. And I need to have a positive control to make sure that I'm testing my assay will work in the presence of intranuclear coccidia nucleic acid. And because of that, the costs, and then if it's a conventional PCR and we always confirm our positives with sequencing, then we need to cut that band out of a gel, send it in to have sequence analysis done. So there's multiple steps in there. And because of that, that's what makes the cost of PCR be a little higher. For human medicine, it's more expensive. You can kind of decrease costs at a high scale. But again, for a lot of the things that we test for, there's kind of a minimum cost we have to meet to pay everything and keep things going. And one of the issues that there is with veterinary diagnostics is if the two of you wanted to start a veterinary diagnostic lab tomorrow and buy a PCR machine and just buy some primers and advertise that you're running tests, you could do that. No one would come in. No one would check to make sure that the test that you're offering is accurate. No one would check and confirm that you're running the appropriate controls. And if you wanted to charge $30 to test for something, everyone could send you a sample for $30, test it, and you could give them all negatives or all positives and nothing. There's no checks and balances on that. Because of that, in the veterinary diagnostic or animal diagnostic market, there are a number of options out there that may not be running appropriate controls. And so that's kind of where I don't know how they do it. I know that we keep our costs 100% as low as we can. Any excess money that we make goes back into research. I spend so many hours a week working in that lab, and I get paid nothing to work in that lab. That's not my job, but it's a passion of what we do. And so as a pet owner, I understand the challenge with if I had a pet and I needed to pay, why is it cost $100 to do that test? But on the backside of things, you can see why it costs $100 to do the test, and it's sometimes a little more complicated. So I see both sides of the coin. I would just say, work with your vets, work with laboratories, let them know situations, and sometimes things can be worked out. But it is my partner. We've had issues with pets before, and sometimes you drop an immense amount of money just to save them, and it's hard. And so I entirely get it. It's one of the challenges of vet med. I wish everything could be free, but unfortunately it's not.
Yeah, right. It's a function of quality. You want the most quality for the least amount of cost. So it's sort of an optimization. Yeah, that makes a lot of sense. In terms of just sort of veterinary diagnostics and the development of the techniques, it's really cool to get your perspective on it as someone that's actually sort of building the molecular machinery that's doing the actual test. I mean, you don't get that. There are only so many people that do that kind of thing. So it's a cool insight. Are there any particular projects that you're working on or that you know of that are just exciting to you in terms of diagnostics and new diagnostics?
So, I mean, I am 100% complete dork for this stuff. So, most of it is pretty exciting. There are always, anytime we're trying to characterize things that are not well studied, it gives me a bit of excitement. I think the Amidomyces story is one that I find very interesting and exciting because, initially we believed it was just this one fungus, and now we know that it's probably more than one, and we're really trying to understand what that means. What does that mean? Where did the fungus come from? What does it mean for health? I'm working on an interesting one. I mentioned an alligator snapping turtle, and that we had, we collaborated with a group at the University of Georgia, and we published on these PIZ2 viruses, which hadn't previously been found to cause disease in any vertebrates, but they caused inflammation in the brain and meninges of these turtles. We had also done a necropsy a year and a half ago on an American alligator, and it had inflammation in its brain. And when we did the sequencing, the American alligator had the exact same virus that the alligator snapping turtle had. And and those cases were like 10 years apart chronologically. So that means that there is a virus in the aquatic ecosystem that those two species share that could potentially transmit between them and cause disease, and we don't know anything about it. There's a number of, there are the viruses that we know in tortoises. We know the herpes viruses pretty well. Intranuclear coccidia, we have a lot to learn, and that's kind of an interesting one to study. Ranaviruses, we know a fair bit, but then there's viruses that we know little to nothing. I mean, the tortoise picornaviruses, they've been documented a couple times. We don't know exactly what they cause, but in working with a friend and colleague, we've now identified those in some really interesting tortoises that may shed light onto the whole subject. I mean, I really love what I do. And so I get excited at most things. And in some ways I have to check myself because, it's sad to get excited about a virus that kills a bunch of animals because I don't want the animals to die. But I'm not inventing the virus. I'm not releasing the virus. I'm just trying to help characterize what's already out there that otherwise would have been overlooked. So, I do find that interesting, and it makes coming to work every day enjoyable.
Yeah, that's it is interesting. We've talked to enough people on the podcast now where you just have all different sort of personalities, and it's all united by the love of the animals, but just different aspects of it. It's definitely a good thing because it's not everyone's sort of interest, and it's good that there are people like yourself that are doing this kind of work. If maybe switching a bit to some more general kind of rapid fire research related questions in general. One thing I wonder about is a lot of times people with captive collections of turtles I am one of these people. I don't have a lot of captive turtles. Being in college, I can't really manage it, but it's sort of, you get into a flow about how you do things and you learn things, but you kind of say, okay, I'm doing it like this. Are there any unintuitive things that stand out to you as detrimental to captive keeping of turtles related to sort of Testudine health that you would sort of say?
When you provided me this question in advance, I tried to think of a couple of options for this one. I think that the thing that worries me most about the captive husbandry of turtles and tortoises and the unintuitive things that we need to think about really is probably more than any other reptile or amphibian we keep in captivity. Where you are located geographically should play a major decisive role in the species that you decide to keep or not keep. Because many of these species, based on the size they get and their UV requirements, can't always be provided with those UV requirements in a captive setting in certain areas. I think that I would really like to see more emphasis placed on keepers of, this is where I live. This is what I should really accept is acceptable for me to keep for the health of the animal. Paul, you mentioned you're in upstate New York. A sulcata or a Galapagos tortoise should not ever be in your future as much as you want it, because there's absolutely no way you can give that animal the UV it needs to be healthy. And that's, I think it's human nature, and I think it's especially nature of those of us that like to keep things. That's a hard pill to swallow for someone to tell you you should not have this because of where you are. I mean, there's a lot of things that come up when we talk about that are major socio-political arguments because of what you should or should not have. And I think probably that's the most unintuitive thing when it comes to turtles and tortoises is, should you keep that species based on your geography, unless you have an absolute endless supply of funds and can create the most incredible artificial habitat ever. There are some species that lend themselves well to it, and then there are others that it doesn't matter what you do unless you build a dedicated place for those. And I think even most zoos struggle with it too. There's just some species that shouldn't be in some places, and I think that in probably tortoises more than turtles, but that's probably the one unique aspect that we don't see in many other aspects of exotic animal ownership.
Yeah, I mean, in New York, I think they crack down pretty hard on even keeping native species. So it's very limited what you really can have without permit. And then there's just the species that you can have that are out of state often don't really acclimate to the New York climate, especially because you're either keeping them indoors, or what's ideal is if you can keep them outdoors. That would be ideal, which most species can't really handle 5 months of inactivity.
Right. And even the places that can. So let's say, we have lots of people in Florida with giant tortoises, right? The temperature and the sun exposure allows them to keep those animals. But I mean, I don't know how much both of you have heard of edema syndrome in giant tortoises. Even in areas where the environment allows you to keep giant tortoises in captivity are living a fraction of the length that they live in the wild. I mean, they're succumbing to systemic disease where they get issues with their heart and edema pooling in all of their tissues, and they're not getting much past 30, 40 years. I mean, we still just don't know enough. Is it related to forage? We're not allowing them to eat correct. Something that we don't know enough about the husbandry. And when you have such a majestic species as an Aldabra or a Galop, and you're cutting your life expectancy to 20%, that's something to think about.
Just on that note, briefly, I remember in the Galapagos giant tortoise book that came out a few years ago, they mentioned that was an interesting thing. It's potentially a cardiomyopathy. I don't know if has there been a lot done since that, or what's the likelihood? Maybe that's a symptom of something else, sort of, that you've mentioned, but is that what ultimately causes the death?
Well, so ultimately they succumb because their cardiovascular system can't keep up, and they're losing lots of fluid into their body cavity. But as to why, there's not really a good feeling. There's a round table discussion paper that's in the Journal of Herpetological Medicine and Surgery that came out a couple of years ago, where all of the people that are really involved in giant tortoise vet med and that talked about it and say what they think might be going on. And the reality is we don't understand. And it's one of those things where it needs a lot more data. It needs a bigger sample size. And, there's not that many people keeping giant tortoises that have been doing it long enough where they have animals that are reaching an age where they're succumbing to this disease.
Right.
Yeah.
Right.
I mean, I see a lot of back to what we were saying just about keeping tortoises and climate is Florida is temperature wise, there's a lot of leeway with what you can keep alive. But Florida is decently humid, and I see a lot of these keepers taking relatively arid tortoise species, stuff like that. All I can think of is that really opens the door to respiratory infections, problems like that. Yeah.
I also remember seeing something a while back about fungal pneumonia potentially in giant tortoises as being a particular issue. I don't know what's the latest on that. I think you see a lot of those cases.
Yeah, I mean, fungal pneumonia is absolutely an issue in tortoises, both captive and free ranging. I do a lot of gopher tortoise necropsies, free ranging gopher tortoises, and we see fungal pneumonia quite a bit. There's kind of a range of different types of fungi that can cause it. Some of them, so that the big fungi that kill reptiles kind of across the board are all in the order Onogenales. So we call them onogenalian fungi. So Ophidiomyces, which is the cause of snake fungal disease, is in that order. Imidomyces, the cause of imidomycosis, is in that order. Nisiopsis, which is the cause of yellow fungus disease, as it's sometimes called, and bearded dragons, is in that order. So they're all really important pathogens. And so sometimes we see them causing the fungal agents. But there's also a lot of other fungi. One of the ones that's the most concerning to me is we see disease in captive reptiles, also free-ranging reptiles, due to fungi that are supposed to only infect insects. And they're called entomopathogenic. And one of the types is Metarhizium, and another type is Bovaria. And they're supposed to only kill insects, but we see disease in reptiles, tortoises included, crocodilians. And the scary part is that those same fungi are used as a pesticide. You can buy Metarhizium and Bovaria from Amazon and sprinkle it across your lawn to potentially kill mosquitoes and things like that. But now we know that they can actually cause disease in reptiles. So what are the implications for free-ranging populations because of those fungi? So yeah, fungal disease is an important cause of disease in tortoises, turtles as well. I would say I think we see it more in tortoises, but yeah, something to absolutely think about. And there again, talk about an insidious disease process. Fungal pneumonias, they're slow. They eat away at more and more of the lung over time. And the antifungal agents that we use in veterinary medicine don't work very fast either. So if you do find a fungal infection, the animal needs to be on drugs for a very long time to try and get rid of it. And even then, it's not always guaranteed it's going to work.
Right. Well, maybe we can cover one last kind of major area before we start to just wind down with a few shorter questions. We've sort of, I think it's actually a good area to sort of wind down and to bring everything together, because we've talked about a lot of different specific diseases and a lot of different aspects of them. But I'm curious, goes back to that paper that we discussed in the beginning of emerging infectious diseases in turtles and tortoises. I'm curious, what are the worst on your radar, and maybe just the consensus about what the worst emerging infectious diseases at this moment in time for turtles and tortoises are, and what worries you the most about them?
Yeah, so I think it depends on, so tortoises to start. Tortoises, hands down, it's intranuclear coccidiosis. I see cases a lot. It's to the point where if I am presented with a dead, radiated, or star tortoise, it is intranuclear coccidia until proven otherwise. That is how common it is, and that is how fatal it is. So with tortoises, intranuclear coccidia is a huge concern. I think cryptosporidium is a growing concern in tortoises. There are a couple different types of cryptosporidium in tortoises. The one that gets the intestines, I think we know a fair bit about, but we're starting to find more about the one that targets the stomach. And I think the one that targets the stomach may be an important cause of hatchling and juvenile mortality that's kind of overlooked. And I would say, for tortoises, those are kind of my two biggest infectious disease concerns. And then for turtles, aminomycosis, the aminomyces infections are really, really important. Herpesvirus infections, we see a fair amount of those in turtles as well. But again, I'm always incredibly concerned about the stuff we don't have good reasons for. Why do some of these species, particularly the ones that are still being imported and not doing well, why are they failing? Is it husbandry? Is it infectious? But those are probably, kind of my biggest concerns on the day-to-day from the cases that I see.
Right. That's interesting. And yeah, with regard to the Testudine, the TINC, where are the biggest unknowns with this sort of this disease, and where it should future research focus?
There is so much we don't know about that one. We still do not have a great idea of who the definitive host of intranuclear coccidia is. There was one study out of Europe that did show that there was sexual replication of the organism in the intestinal tract of a leopard tortoise, but that is the only study that has ever shown that. Until we can culture intranuclear coccidia, we're limited in what we can do to truly understand it, because unless you have the organism in culture, you can't do additional studies. And so that's an area where we need to get the thing in culture. And so I have plans to try and do that in cell culture. Like I mentioned, I've got a lot of different tortoise and some turtle cell lines. We're going to try and get infected tissues, put those tissues on the cell lines and see if we can get the parasite to infect the cells in tissue culture. And if they can do that, then we can grow it, and then we can do lots of important things. Where the organism tends to hang out before it causes systemic disease, I think, is a really important question. We don't know that very well. What makes there be outbreaks in certain situations? Sometimes collections will be completely fine, and then all of a sudden it'll just pop with intranuclear coccidia. It'll go through like gangbusters and kill lots and lots of animals, but it's unclear how it got introduced. Was there an individual that was serving as a reservoir? And then we need better drugs. Panazaril is a good drug to decrease the parasite load in animals that are infected, but it never clears them. And if you can't clear the animal of the infection, that means you're either treating it for the rest of this life or with the drug, or you're just waiting for a time at which it becomes stressed for the organism to bounce back, start shedding again, and put the rest of your collection at risk. So it's a really challenging organism, and there's a lot we need to figure out.
And so the fact that it's intranuclear is an interesting. What are that's not a typical thing you see, from my understanding. And what are the implications of that aspect of the organism, the Coccidia's life history, that are particularly damaging?
Yeah, so we don't, Coccidia as a group generally infect epithelial cells, though they can infect other types of cells. And most often we see them in the cytoplasm. That's kind of where they replicate. There are intranuclear coccidia that have been described in other species, but I would say it's not a thing that we commonly see as a cause of disease. And it can make diagnosis hard, because the nucleus of a cell has a lot of chromatin. That's where all the nucleic acid is. And it's a kind of an easy place for organisms in low numbers to hide. And so, when I first started, so when I wanted to first see my first intranuclear coccidia case was in my residency, and it was recently described. And some of the tortoises in that study came from the Bronx Zoo where I was studying. So I went back in the archives and I pulled the slides of those first tortoises, and it is a hard thing. You can see all this disease, but seeing the organism is really, really hard. And so, it took me a little while to actually learn to be able, because all the only information out there was a couple of pictures in the first publication, but that's not enough to really become adept at diagnosing it. And so, it can be really hard to find. Sometimes I have a case and I'm like, I know that this is intranuclear coccidia, but I'll spend a long time searching for the freaking organism. And, then I'll do PCR and sometimes it'll help, but there are still some times when I'm not ruling it out. But then there are other cases. I mean, we had a group of little baby star tortoises that someone bought out of Craigslist. They thought they got a great price for them. They all dropped dead in a few weeks. And these baby star tortoises had more intranuclear coccidia than I had ever seen. Like I'm attending a conference for a regional pathology conference next weekend. That's the slide I submitted to that conference, so I could teach everyone what this thing looks like in a case where it's all over the place, and then tell them, okay, now you need to search sometimes in your tortoise cases is for a long time, because this is what you're looking for. But that's one of the, we talked about things like if we had an antibody to this, we could then make a special test where we could stain tissue slides quickly and diagnose it more accurately. And so, but we need to get it in culture to do that.
Interesting. So the tough part of seeing it, is it, what aspect, is it just it occurs in low abundance, or is it super small, or is it kind of all the above?
All the above, where it's in the nucleus, low abundance, very small, hard to see. And again, you can see systemic disease, and the distribution of the organisms is not consistent. Sometimes you always want to look in the kidney. Sometimes you always want to look in the pancreas. Other times it's in the lungs and the brain, and the other organs don't have anything. So we don't know enough about that bug just yet.
It's fascinating. I mean, it can make sense why it hasn't. It sounds like, yeah, trying to isolate the host for the organism would be the logical step, and that makes the therapeutic side to the diagnostic side much, much better, right? But that's going to be tough, right? Because it probably is the same situation. Maybe someone may have looked at a slide with the host and just didn't know what to look for, if they were doing some other.
Why are so many tortoise species really, really susceptible to this? But then only a handful of turtles seem to be. So why is it infecting some turtles at all? Like there's something weird about this organism we don't understand yet.
Yeah, well, it's I mean, this is the epitome of the detective work behind this. It's really interesting stuff, and like a cool puzzle that requires a lot of different lines of reasoning and collaboration. It's really interesting. I mean, yeah, and just I guess to wrap that up, because it's sort of I think an important topic and an interesting topic, and because it's so unknown. I guess for someone that could have that issue in a collection or something like that, what would you recommend is the best thing to do to get it diagnosed, or if it is diagnosed, how to offset it, sort of stave off the impacts?
For diagnosis, PCR is really the best. And the good thing is that there is a quantitative PCR for intranuclear coccidia. So that means that is a test that only detects intranuclear coccidia. It doesn't require sequencing, so the turnaround time on that test is faster, and the cost is also a little bit lower because it doesn't require sequencing. So we know that cloacal swabs or feces is our best sample to detect it, probably in most species. But if you have tortoises with a respiratory disease of unknown origin, a oral or choanal swab would also be a really good thing. As for what to do when you have it, I think this is one of those diseases that, unfortunately, with what we know right now, you need to make really hard decisions. Is this an animal that is of enough sentimental, genetic, or personal value that you want to work on treating it with antiprotozoal drugs, and you are probably committing at least intermittent treatment for the rest of the life of that tortoise based on what we know? Or, are you going to then maintain, some people can maintain. I work a lot with snake serpentoviruses. And so we see a lot of this in pythons with serpentovirus infections where, do you maintain a clean colony and a dirty colony? Like, these are animals that are important. They have the parasite, but you don't want to mix animals that are negative with animals that are positive, and you hope that the animals that are positive just live out a long life. Or is it an animal you need to unfortunately consider culling, particularly if it's a very susceptible species, to then factor in the health of your other animals and your interests in the collection? If you are a commercial, or you're breeding for profit and you're breeding to produce animals to sell, this is not something you want to have near your breeding stock if you're selling babies.
Yeah, interesting. One more thing too, I from the roundtable discussion paper on the coccidia, I seem to recall that there isn't. It's assumed that it's within the family Amaridae, but that it's not fully understood exactly what the organism is. Is that, you'd think that, is that just because there aren't enough sequences in GenBank or whatever to compare to the sequences you have from the PCR, or why is it that that you haven't had a specific, been able to diagnose exactly what it is?
Parasitology as a field is very based on old school techniques of morphology. Because we don't have it in culture, we cannot determine the culture characteristics of the parasite. And therefore, without that culture characteristics, you can't assign it with just genetic sequence alone. That is not accepted in the field of parasitology. So until it is cultured and then someone can look at those developmental stages of the parasite, it cannot be officially, like, we could give it a name in the next paper, but it would never be accepted by the field of parasitology. So until it's in culture, we can't be 100% sure, but we can, at least with our genetic sequences, have a rough idea.
Okay, that makes a lot of sense. Well, yeah, I mean, it's really interesting to get your perspective on it as someone who's really working the front line of characterizing that and many other sort of issues and diseases. I think we can start to sort of wrap up. We'll finish with a few kind of broader questions. And I mean, one thing that I am kind of interested in, just in terms of your adventures, those kind of things. In 2018, I had the opportunity to go to Madagascar for the big tortoise confiscation. And so got some really, I mean, I was in 8th grade at the time, so got some really incredible experience working and seeing with Dr. Raphael and a lot of others from all over. And that was really interesting. And I know that you've had a similar experience working with the Burmese star tortoises. And potentially other things that I'm not aware of, but what was it like from your perspective? As a kid, I was watching a lot of the vets there, and I was thinking this is really cool, but there was a lot of stuff going on, in terms of getting the samples they needed. What is it like to do that kind of work in another country? And
And the thing is that as a kid, you said it was really cool. As a vet, we're going, oh, this is really cool too. It's working in another country was really, I've done some international work, not a ton. The Burmese star tortoise project was really, really unique in that it's fantastic how much success there has been in the captive conservation of that species in these conservation colonies. But you absolutely want to make sure that the animals that you put out in the wild aren't carrying any potential diseases that could infect either remaining Burmese star tortoises or other, Burmese brown mountain tortoises, other tortoises around there. So you don't want to put anything out that has any disease. So, with them being, a CITES species, it is really, really hard to get any sample out of the country and into somewhere else. So it was much easier, and believe it or not, cheaper, to send a team of people and all of our PCR machines to Myanmar to do the testing in Myanmar. And so, we packed up all of our equipment. We, flew it. You have to make sure you've got everything to do all this testing. Fly it to a country like Myanmar where you're not 100% sure your high dollar equipment's actually going to even make it through the process without being confiscated, damaged, stolen. And then, some of these star tortoise facilities, I mean, they were super rural areas. I mean, we were doing our PCRs in one of the locations. It was an amazing place. Lots of really cool people. They had a generator for us to start to run our PCR machines. We're doing microscopy by headlamp under mosquito hoods in the middle of nowhere. And then the generator goes down. And that's our only source of doing this stuff. So people contacted the individuals in the native Myanmar people, the Burmese contacted people the next village over to see if there was a generator that we could use. And they did. They had one, but it needed to be brought by ox cart, and the ox were need to work in the fields during the day. So we got the generator in the middle of the night the next day because they needed to bring it by ox cart in pitch black to get it set up. I mean, it just, all the things that you take for take advantage and take for granted in the lab setting, they're not there. And it's amazing. I was most impressed. You see these people and how just incredibly dedicated they were to the conservation of this species in their own country and how important it was. And it makes all the work, I think, that I was doing worth it. So.
Yeah, I mean, that's really an interesting point. I had a similar, just to see people in Madagascar that are living on $70 a year. I mean, it's that ridiculous. I don't know exactly what the case is in Myanmar, but I'm sure it's similar. And to see how dedicated they are and how happy in general, they were just very, it's like it shows that there's certain things that you don't need money to be really happy and enjoy things. That's cool. In terms of, I'm always curious, specifically with vets, what's the most interesting case, maybe turtle or not necessarily turtle related, that you've ever worked on in any capacity?
I mean, I guess the most interesting cases are always the species that I've always loved. The first adult galop necropsy I got to do was just unbelievable. My first Komodo dragon was, so cool. My first big, like real big American alligator. I think it's really all the firsts, or it's the species that, are particularly of interest to me. I mean, the first time I got to do a Eastern indigo necropsy was, something else. So it's all the super cool. The first time I got to see Kahansi spray toads that were destined to be released in Tanzania and see them on a glass slide. And, they're a species of frog that keeps the eggs after they're fertilized within their coelom. And so you look at a slide of a female Kahansi spray toad and she's got little tiny froglets inside of her coelomic cavity. There's so much cool stuff out there.
That's awesome. Yeah, just the little things, that wouldn't necessarily make it into sort of an academic paper, and just the personal takeaways. Something that we touched on a little, and maybe we can just kind of come back to it briefly, is you've mentioned sort of that there's between just veterinarians and maybe in general and public some distrust when it comes to reptiles, that sort of thing, or a little tension. I'm curious, what do you think causes that, and what are the ways that going forward we can strengthen the trust and the connection there?
I think there are things that probably could be better handled on both sides of the aisle. Starting with the veterinarian side, I think one of the challenges that people need to kind of get over is, and I understand in part where it comes from, is you as a veterinarian likely are not to know as much of the captive husbandry and biology of the species you're seeing if you're an exotic veterinarian than the owner. Many people, especially those who are keeping the more uncommon species, are incredibly well versed in the animals they work with. They spend their free time reading a lot about these animals. They know what the natural habitat is. There's no way a veterinarian is going to know that, and a veterinarian needs to be okay with that. And that's kind of a 180 from where they would be on a domestic animal or a farm animal. Because a veterinarian many times with a domestic animal or a farm animal is dealing with owners telling them all the things they read online that are from someone who probably has no training whatsoever, saying it, this is good for my dog, this is good for my dog, this is good for my dog. So they have to change the mindset from that to, oh, wait, this owner knows what they're talking about. I don't know that. And I think a lot of veterinarians struggle with that. So veterinarians need to be more accepting and open to the idea that they don't know everything about all the animals they see, but they do understand disease. And I think the one thing that, maybe owners can help with in a little aspect of that is, just because you're working, you're keeping an uncommon or atypical species, if that veterinarian has never seen one before, or if that veterinarian pronounces the name of the species wrong, and I get it, if you work with this species of animal your entire life, and you see a vet who's supposed to help your sick animal and they don't even know how to say the name of the animal you're bringing in, that's a little off-putting. But they understand the disease mechanisms that may be going on. So they don't have to have necessarily seen that species before to be able to help you in that regard. So I think it's kind of maybe decreasing the barriers and those protective mechanisms on both sides of the coin would probably help the interaction a lot more. If everyone just accepts that, it would move a little bit more smoother.
Right. Yeah. Be open and recognize that makes a lot of sense. Right. It's like, yeah, from the sort of patient side, it's like, yeah, they have training in the dynamics of how the disease works and sort of, but maybe in certain cases, I mean, the state sample space that you work with exotics is so vast that, I mean, you can't expect someone to know every detail about, from the veterinary side, about how that animal should be kept, and relying a bit on that information, there could be a really positive dynamic there. That's a cool thing.
I think with that like level of understanding, like an example is I removed a African side neck turtle that I found in a pond up here, and one of my buddies who's not even really a turtle guy is keeping it now, and I would honestly genuinely say he knows vastly more information about African side neck turtles than I do. And there's just like that understanding of when you keep an animal, you start to specialize in it, and going into a veterinarian setting, there needs to be that understanding that where you stand with that animal and what they know.
Yeah, it's cool. I mean, a lot of people I know that keep turtles, and with dogs and cats, it's easy to Google and you'll get the 5 top tips you need or whatever. And who knows if that's reliable, but for a lot of turtles, you have to go straight to a journal article. That's all that exists. So it does take a little bit more specific type of personality and interest in that. I think something else too is just like, there's this in any field of science, I feel like there's this unfortunate just reality that the people like yourself that are at the front line of sort of discovering things and you're busy, and it's tough to be able to communicate and find the time for it. And it's just good to open the dialogue, and we have these outlets like the podcast and that kind of thing, and just to be able to bring this to people from the source and the people that are on the front lines. It's a really beneficial thing. So we're really grateful that you've taken time out of your schedule to talk with us, Dr. Ossiboff. It's been a pleasure. And I guess the last question we like to end with is just, if you've got one piece of advice for someone looking to make turtle and tortoise research or veterinary work a career, what would that be?
Don't give up. I mean, it is find your niche, right? There's so much in regard to these species that we need to work on. Find something that excites you about that species and go for it. It's you're going to have to have some flexibility. You're going to probably have to have flexibility in your life plan. You're probably going to have flexibility in your geographic location where you end up, and you're going to have flexibility in what your finances are going to look like. But it's rewarding. And, I think in the end that for a lot of people, just being able to help and make a difference is absolutely worth it. I know it is for me. And I mean, I love doing, I can't do as many of these things as I would love, but I've had a great time sitting here and talking with you too, and talking about this stuff. And hopefully people are going to listen to it and enjoy it. And that's, if you love this stuff, find a way to do it. And I think that it'll be better for the future. It'll be better for the animals, and it'll be better for everyone who works with them.
Awesome. Yeah, we will take that to heart. And for people listening, I think that makes a lot of sense. Thanks for the advice. The last thing we like to do too, we do this at the end of every show, is like a quick round of turtle trivia or random veterinary turtle related trivia. I don't know if you've got some just wild fact or something that would be useless anywhere else, but that you could throw our way and see if we can.
So the one that always comes to mind, and I don't know why, but so the male turtles have a phallus, right? If you've ever seen the phallus of a male soft-shell turtle, It looks like the Demogorgon from Stranger Things. And it is the most it has all of these little, like you never would expect any, it's just, yeah, look at a picture of a male soft shell turtle phallus and you will see it is the craziest thing ever. Even people that do this, what I do routinely, someone I was around, she does necrop shield, she said, "What is that thing?" And I was like, "Yep, that's the soft shell phallus." So I don't know why I decided to end up with a phallus trivia fact, but that's all I had.
It works. We've had all manner of them. So but that's a really good one. There's a lot of, that they are pretty crazy. In the Colonial Research Institute collection, Dr. Pritchard had a series of jars of turtle phalluses. And there were a few soft shells in there at least, and it was just that was part of the collection, which it's useful, I guess, to some extent. But yeah, it was just kind of, you never know what you were gonna see there. It's just incredible. Yeah. But all right, well, thank you for talking with us. We've been planning this for a while, and I'm glad it all worked out. So that was awesome. And
absolutely.
Thanks for the work you do.
Yeah.
And
if anyone out there, you can find my website at UF. I try to respond to every email I get. It may take me 24 hours or so. Shoot me a line if you're having concerns with animals dying. Absolutely reach out to me. Necropsies, they cost some money, but we try to keep our prices super cheap so we can help people and learn from these things. And my job is to help everyone out there working with these animals. So if you need me, just reach out.
Awesome. We can link it in the description too of the video for people, and we'll have a link to the website and also the ARAV website. I think that would be good.
Absolutely.
So for everyone, you can find that there. This has been episode 55. Thanks for joining.